Hello, and thank you for joining today's webinar. My name is Jacob Parnell. I'm an agronomist at Biome Makers, and will be moderating today's webinar. For a few housekeeping items that I'd like to bring up, this webinar will be recorded, and a copy will be emailed to all of those who have registered. Please, as we go through the presentations, as we go through some of the discussions, use the question panel for any questions you may have during the session, and we will dedicate time at the end of the webinar for answering those questions. And if we don't get to them, we will reach out through email to make sure that those questions are answered. And then finally, at Biome Makers, we believe in sharing our knowledge and our BeCrop technology with the world to further our mission in recovering soil health. We offer free certification for biological soil analysis called BeCrop Advisors. And for anyone who's interested, we'll be sharing more information on this in a follow-up email. So I'd like to get us started by having our panelists, David and Fatma, introduce themselves. David, would you like to go first?
Sure. So I'm David Shapiro-Ilan. I'm the research leader at the USDA ARS research station in Byron, Georgia. I studied biology at University of Michigan, then spent 2 years as a Peace Corps volunteer in West Africa and Niger. Obtained my master's in entomology from Louisiana State University, a PhD in entomology from Iowa State University. Then I was a Fulbright postdoctoral scholar at Volcani Institute in Israel, and subsequently spent 2.5 years in industry researching and implementing mass production of entomopathogenic nematodes, one of the organisms we'll be talking about today. I joined USDA in 2000 and became research leader just a few years ago in 2019. The focus of my research is on invertebrate pathology, invertebrate diseases, and biocontrol of insects using microbial agents. I've published 220 refereed papers on this area, as well as 26 book chapters in 4 co-edited books, and I'm an inventor, own 7 patents. I'm a fellow of the Society of Nematologists and of the Entomological Society of America. I was previously editor-in-chief of the Journal of Nematology, and now I'm serving as editor-in-chief of the Journal of Invertebrate Pathology. So that's me in a nutshell.
Well, great. Fatma, would you like to introduce yourself?
I'm Fatma Kaplan, CEO and founder of Pheronym. We are using a new kind of pheromone to control agricultural pests, and our application is beneficial nematodes, the first application. And I have a PhD in plant molecular and cellular biology from the University of Florida. And after that, I did a postdoc at the National High Magnetic Field Laboratory to identify mating pheromone from C. elegans. And we published this work in Nature 2008. Then USDA hired me to apply this new finding to agriculturally important nematodes, specifically root knot nematode. We initially thought about using the mating pheromone, and we quickly realized that root knot nematode didn't mate to reproduce and didn't need to. Then we had to look at some other ways to bring this new finding to control agricultural pests, and one of the behavior was dispersal behavior. I actually read David's publications about the dispersal and the problem in beneficial nematodes. Of course, I didn't know David at that time. And I looked at this behavior, whether it is actually regulated by pheromones, and when I found that, we published it in 2012. And we thought this behavior can also be applied to root knot nematode. That's basically seeded the Pheronym, the company currently we are developing the technology. In 2015, we established a CRADA with the USDA and David's unit, and from there, we have been developing the technology, and we were very happy to do this study with you and USDA.
Well, thank you, Fatma. My name is Jacob Parnell. As I mentioned earlier, I'm with Biome Makers. I'm an agronomist. My background, I have a PhD from Michigan State University in soil microbial ecology. I did a postdoc and subsequent research faculty at Utah State University in their ecology center, focused on microbiomes, and then I had a 3-year appointment with the National Ecological Observatory Network in Boulder, Colorado, designing the terrestrial microbial ecology system. After that, I spent 7 years in industry working on developing biological inoculants for plants, and am currently at Biome Makers, as well as I have some affiliation with North Carolina State University, studying plant microbe interactions. I'd like to give a little bit of a background of Biome Makers, some of the history, kind of an introduction, and then I'll kind of give a little bit of a, David an opportunity to give a little bit of the technology as well. So our topic today is what can entomopathogenic nematodes do for soil health? And soil health is something that has been an issue that's been hard to define. Everybody kind of knows what it looks like, but they don't really know how to measure it. And so that's one of the things that we're working on at Biome Makers, is really trying to take that the aspect of soil health and quantify it, give it some measurements that we can really look at to determine what is soil health. Biome Makers was founded in 2015, and it was really designed to look at the microbial communities as kind of almost like a canary in the gold mine type of, these are the ones that are going to be most affected by anything that's happening in the soil. We should be able to see shifts in the microbiome that help us understand better what is going on in depth in the soil. And we're really driving this to build healthier soils, to build more sustainable agriculture, and to enable farmers, input manufacturers, and retailers to be able to make smart decisions based on actual measurements. And so this is kind of our team. It's a rock star team. We are a worldwide team. We have labs in several locations in United States and Europe. Our US headquarters is in California. We have 70 plus employees, top scientists, top everything. It's a very dynamic and very interactive team. And across all of this team, we've got over 150 years of experience in agriculture, in microbes, in sequencing, in the sciences. And our impact, as I mentioned earlier, we're really driving towards sustainability. All of us at one point started off thinking, we want to change the world. And so that's really kind of how Biome Makers is looking for these innovators, these scientists to bring on board, is that passion to change the world, to make the world a better place. And so just through our technology, just through making these smart decisions based on science, we have been able to make huge reductions in the amount of agrochemical fertilizers, because once we have growers that understand the biology, they understand better what inputs need to be made and maybe what inputs don't necessarily need to be made. We also see that because of this, we're able to help with management practices on some of these farms and these acres. And so because of this, we have increased the carbon sequestration in agriculture by 15%. We're growing quickly as far as the outreach that we're able to do, with over 8,000 farmers becoming more regenerative and looking for ways to help quantify what they're doing and to be able to show that they are making a difference. We have 300 people that are already these BeCrop advisors that are trained people that are freely trained by Biome Makers to be able to use these tests, to be able to look at the biology of soils and to be able to give interpretation and maybe some direction as far as application and management practices that can be used. We have one of the largest taxonomy databases in the world based on all of the thousands of soil samples that we have collected. And we continue to build, we continue to grow, and have contributed not only on the application side to the growers and to input manufacturers, but on the academic side as well. We have a lot of university partners and partnerships, and we have been able to contribute to several scientific publications. Overall, we have over 200 industry collaborators, and our products, our testing is done on over 63,000 hectares of research and development projects worldwide. So I want to kind of go back and kind of give a little bit of analysis or a little bit of background about what exactly it is we do. We're really trying to describe the life that's happening inside the soils. Each gram of soil has millions, if not billions, upon billions of microbes. And there is an ecosystem there, and the plants are basically growing in this ecosystem of microbial life. They're helping with nutrition cycling, they're helping with disease resistance or pathology, if that's the case. And so they're really playing a very dynamic role in plant health. And so we like to use these as kind of indicators of what the plant can see as far as those nutrients, the nutrient cycling, as far as stresses that are going on below ground and that are affecting the plant above ground. So we really look at the biodiversity of those ecosystems. What does that biodiversity look like? Plus the functionality, or what are those microbes actually doing in the soil to help benefit the plant or the soil itself? And overall, we're able to encompass these into actual measurements of soil health. So we're able to give indices that provide metrics of soil health itself. I mentioned a little bit about this database that we use. What we do is we collect a soil sample, or our collaborators that we're working with will collect a soil sample. They'll send it to our labs. We have several labs all over the world, and there we'll extract the DNA. We will sequence the 16S and the ITS regions of the DNA, and then we'll take that and compare that with a database that we have on the full genome sequence. And based on the changes of the abundance of microbes, and then referring back to that full genome sequence where we understand the function of each one of those microbes, we're able to look at how the function, that potential function, changes over time. And so we really have kind of a big kind of functional and ecological approach to soils and to life in the soil. And we are, right now, we have done over 122 crops, 40 plus countries. We are really trying to go worldwide. We are really trying to bring the idea of soil biology to the forefront, to make it an integral part of agriculture's understanding the biology of the soils. So as I said, we start with the soil sample. This is usually collected by a consultant or a grower or the input manufacturers as they're doing their field trials. And then they're able to send that to us, and we're able to identify these biological pathways that are involved in, yield, nutrition, stress, adaptation, management practices, whatever they're looking at that might change the biology. The technology that we have is very fluid, and it is able to measure a whole bunch of different parameters. If it's changing the soil health, we can measure it. And so we've done a lot of management practice changes. We've done yield differences. We're even getting to a point where input manufacturers are able to do large scale field trials, and we're able to show and recommend where their product will work based on the biology. And in the end, what we want to deliver are some reports that are usable by those collaborators that we're working with, so that they're able to deliver it either internally to their R&D teams, to commercial teams, to be able to say this is what our product does, to be able to take it out to the community, to be able to say this is what this type of management practice, this is how it changes the biology of the field, or even to the growers themselves, to be able to say these are some of the things that I can do to improve the biology in my soils. And so it's really designed to equip each one of these different entities, these groups, with actionable insights that are based on the biology and the microbial insights of the soil. I think we're going to kind of skip this for now, because we'll talk a little bit more about how we designed what we did for this project. And so I'll turn the time now over to David to give a background into kind of nematodes and entomopathic nematodes.
Let's see, can you guys see my screen now? Yes?
Yes, I can see your screen.
Okay. Is it in presenter mode, or is it just in the?
I'm still seeing it in.
Okay.
Oh, there we go.
I think, yeah. All right. So yeah, as Jacob said, I'm going to just kind of give an overview on these entomopathogenic nematodes and what the pheromones can do in terms of enhancing their pest control or biocontrol efficacy, which is kind of where I came into this collaboration. So I'll be giving it from our research program perspective. So basically, if you're not familiar with these entomopathogenic or insect-killing nematodes, they're potent bioinsecticides. They are commercially available. At least about a dozen species have been commercialized, but they exist more than 100 species described. They fall into two genera, the Stauninema and Heterobditis. Basic life cycle is the infective juvenile nematode, which is the only free living stage, enters the host through natural openings, releases a symbiotic bacteria, which is the partner to those bacteria, and together they kill the insect. The insect dies within 48 hours, and then you get tens of thousands or hundreds of thousands of new nematodes coming out ready to kill new insect pests. So there's a number of attractive qualities that have made these nematodes really important as biocontrol agents. They kill a wide range of hosts relatively quickly, especially compared to other pathogen groups. They have this durable infective stage, the IJ or infective juvenile Dower stage, makes them amenable to mass production and application, and they're extremely safe to humans and other non-target organisms, so much so that they are exempt from EPA pesticide registration and have minimal or no regulation in other countries as well. So these nematodes have been used and are being used against a wide variety of economically important insect pests, such as your citrus weevil, black vine weevil, fungus gnats, mole crickets, various white grubs, various borers and weevils, including pecan weevil. So a lot of success, but at the same time, field efficacy can be variable, and it's sometimes difficult for the nematodes to compete against other control tactics, such as chemical insecticides. So it's important to try and improve the biocontrol efficacy. Nematodes rely on finding the insect host and infecting that insect host. So if we can enhance those traits, infectivity and host finding, we can improve biocontrol success. And that's really what the pheromones do, is improve these characteristics to make the nematodes better. And I'll going to talk about a little bit how we stumbled upon that. So it started for me back when I was doing a postdoc way back in '96 in Israel, when we noticed that if you apply the nematodes directly from these dead insects that have the nematodes growing inside, as opposed to spraying them in aqueous suspension as they're normally applied, the nematodes coming out of that dead insect do much better than those that are applied in aqueous. They do better in terms of dispersal and infectivity, and more recently we found an environmental tolerance. And we saw that translates into higher efficacy if you apply the nematodes in the dead insects, the cadaver, instead of applying in aqueous, and that's what this graph shows. But we didn't know why are the nematodes better in that cadaver than they are when you apply them in aqueous. And in another study where we're looking at group foraging behavior, we like to look at how do these nematodes navigate and find a host in this giant ocean of soil. And we know, of course, that many organisms, including us, display various group behavior. So we wanted to see, do the nematodes also display group behavior when they infect and when they move? We expect that they would, because one tiny nematode against the Goliath insect with its host defenses is going to do poorly. But if they can attack together and move together, then they're going to do a lot better. So we did indeed find the nematodes infect together and they move together. They move continuously through the soil. We think kind of like a pack of wolves, and maybe there's this alpha nematode leading the pack. But again, we didn't know why they're moving together and how they're moving together. And this was published in International Journal of Parasitology, led to other studies and a joint USDA grant that we got with Fatma and other partners. And with that, we've more recently found that the nematodes follow a leader. They do trail following. So if you put one nematode, say, on an agar dish, and then remove it, other nematodes will follow it. So they have this following behavior. And again, what's behind this? So we find, and this was something, and how Fatma and I came together, is when she discovered that these pheromones regulate the entomopathogenic nematode behavior, enhancing, as shown here, the dispersal of the nematodes. And then we found subsequently that the nematodes also regulate infectivity of the nematodes. So you can see this is number of nematodes invading an insect host. It's higher when the pheromones are present and exposed to the nematodes. So if you have these pheromones that are directing nematode behavior, we want to see if we could leverage that to improve biocontrol, so we could tell the nematodes it's time to go out and infect and find an insect and infect it. So that's what we started testing, is can we use these pheromones plus nematodes to enhance biocontrol efficacy? So of course, the overall goal of my program is enhancing sustainable methods of insect pest control. But in this project, we're exploring the benefits of pheromones plus nematode combinations for improving insect pest control and developing technology to implement that. So in the lab and greenhouse, we saw very quickly that if you expose the nematodes to their ascaricide pheromones, you can enhance infectivity. This is percent survival of pecan weevil, a major pest in pecans, and survival is reduced even further with the pheromone than with the nematodes themselves. EPN is entomopathogenic nematodes. So we see this result with pecan weevil as well as black soldier fly. And again, this is greenhouse and laboratory. So we want to see, can this work in the field? The first field test that we did was against peach tuber, a major pest of stone fruits, including peaches, plums, cherries. We applied the nematodes with, and this is another nematode, S. carpocapsi all strain, with and without pheromones, with a relatively low rate of the nematodes per tree. And this insect pest bores into the roots, so you're applying to the soil. It's basically a soil pest. This is a low rate of nematodes. Probably costs, depending on which company, maybe only $10 per acre, which is very low. So we have this low rate of application, but even at that low rate, we saw excellent control of the nematodes with or without pheromones. So we really didn't have room for improvement with the pheromones. So this is not a good pest for this particular test to test the pheromone, but it did show once again how great the nematodes are against peach borer. And it's important to note that chlorpyrifos lozban, which was the recommended pesticide at that time for peach borer, the nematodes did just as well, and we see that time and time again in many field experiments. The nematodes can do as well as the chemical insecticides. Of course, chlorpyrifos is now removed from use. So there was a lot going on besides what we're seeing here in terms of the nematode efficacy, and Fatma is going to be talking about what we saw in the microbiome based on these applications. But what we were after as well was to be able to demonstrate if the pheromones could enhance field efficacy, not just in the lab and greenhouse. So more recently, we applied in a pecan weevil setting. Now, pecan weevil, a major pest again of pecan, in the adult stage is very susceptible to the nematodes, but as a larva, it's fairly resistant. So you can see here in this application made last year in December, Steinema feltii didn't quite control the pest. This is percent survival of the weevil, but nematodes with pheromone didn't. That's especially interesting done in December when nematodes don't usually do that well when it's that cold, but the pheromones enhanced their movement and infectivity even under those conditions. So this was the first field trial where we're seeing enhanced biocontrol with the nematodes and pheromones. So that's really where we came from. We're seeing the pheromones enhancing the nematode dispersal, infection and invasion, biocontrol efficacy increased in the greenhouse and field. We're doing additional field tests underway to optimize and scale up the pheromones with our partners at Pheronym. And as you'll hear from Fatma, these pheromones also positively enhance the microbiome. So that's my presentation. I'll be happy to answer questions a little bit later. Thanks. And I want to acknowledge real quick technical assistance, co-investigators, and funding for these projects. So now I need to stop sharing.
Yeah, so if I can get the screen back, I will.
There we go. All right.
And this next part over to Fatma.
When we started this project and as we got closer to commercialization, and particularly after successful greenhouse trials, one of the questions came to me fairly frequently: what about the effects in the soil? And this was something really critical for the new product development. And even though the pheromone we treated, the beneficial nematodes were very small, and we were able to see a difference that the pheromone treated nematodes were very effective. And by the time we put these nematodes in the field, the pheromone concentration was very low. But this was a question we have been asked by many partners and the field farmers, and we thought we had to do it. And thanks to one of our board member, Pam Marone, she recommended Biome Makers, and she made an introduction, and we talked to Biome Makers how we can actually accomplish this, because we didn't really have expertise in soil microbiome. And at the same time, I reached out to David and I said, well, here is a question we come up fairly frequently. can we do a soil microbiome while we are doing the field trials? As David mentioned in the first the field trial with chloropyrifos, we wanted to know first the residual pheromone level in the field would have any effect to soil microbiome, because it was a very tiny amount. And at the same time, is there a difference between chloropyrifos versus the nematode treated with the pheromones? We expected actually not much difference from the nematode pheromone treatment, and then all of a sudden we had this really surprising positive results. And before we got really too excited about it, we thought that we should test this experiment one more time. And the experiment that was done in the spring was a very nice and warm weather. It had a lot of rain. And the experiment today will be presented is the one with more replications, and at the same time it was in the fall, and it was in Byron, Georgia, not much precipitation. Let's move to the next slide. So briefly, at Pheronym, we are using pheromones to control agricultural pests. And our first product targets beneficial nematodes to improve efficacy, and our second product targets to control plant parasitic nematodes. And one of the major difference I want to emphasize is when people hear pheromone, they always think about the pheromones for the insect pests, and they don't actually think about the pheromones from the nematodes. The pheromones we use from the nematodes are water soluble, and our applications are for the soil pest. Next slide. So after our exciting result in the spring, and before we advertise these exciting results, we thought we should try it one more time. And one of the really big question left with the first field trial, was we were able to see a really nice positive effect with the nematodes and nematode plus pheromones. And we weren't really sure that nematode plus pheromones had that really great effect, whether it was just a synergy between nematode and pheromones, or the pheromones actually, that really small residual pheromone got into soil, had a positive effect. So we talked to David and Biome Makers how we could accomplish this experiment. And one other thing we realized in the first experiment is after 4 weeks, we didn't really have any effect. So everything went back to normal. So this time we did nematodes by themselves to see nematode effect, nematode plus pheromones to residual, and the pheromone by themselves to see actually whether we had any pheromone effect. And then many of the other questions is the synergy, is it a synergy, or how does this nematode pheromone effect happens? So we had in this one, T0 was before the nematode or any of the application. What is the baseline? Then we had the T1, that is 2 weeks. T2 was 4 weeks after. For each experiment, each treatment, we also had 8 samples. We have learned that with the field trials, you need a lot of replications. And with microbiome, we also learned that we had a lot of, and we wanted to see differences, whether they were a statistically significant difference. And we provided the pheromones to David, and David's lab collected the samples according to the suggestions from Biome Makers, and they were sent to Biome Makers, and they have done the analysis. Now let's hear from Jacob for the analysis.
Thank you. So first, the one thing that I wanted to go over is basically some of the alpha diversity. There are 2 different kind of ways that we look at alpha diversity. One is richness. So that's just the number of species that you have in the community. And here I'm mostly in this slide focusing on 16S alpha diversity. So this will be bacteria and archaeal diversity. And here you can see this top set of figures is our richness. And the way we've divided it is we've got the pathogenic nematodes only, and then these are divided up into the Feltiae on this side, this SF is the Feltier and SC is the Carpocapsae nematodes that we're looking at. So we're looking at two different types of nematodes and the pheromones. And then, as Fatma said, we're looking at the nematodes alone, the pheromones alone, and then the combination of the two. And the thing that I want to kind of point out here, so the top figure is the richness, the number of species that we see, and the bottom number is the distribution of those species, so the evenness. So how, is there just one dominant species, or is it kind of a nice distribution of different species? And the one thing that I want to point out here on the bacteria side is that we see that there is, at this time one, we do see that there is a significant difference between the control and all of these, especially in the evenness. We see it between the three treatments that we looked at. So the nematodes alone, the pheromones plus the nematode, as well as the combination of the two, we see this drop in diversity, and we see it with both of these nematodes. But by the time we get out to the 4-week time point, that's T2, we see that in both of these cases, it's no longer significant. So there's kind of within the first 2 weeks, there's a little bit of a drop in this alpha diversity, but by the time we get out to 4 weeks, that has already kind of gone. We no longer see that difference in diversity. When we look at the fungal diversity, we see something a little bit different. Again, I've got them the same way, where we've got the richness, the number of species on the top, and then both that evenness and the richness combined are down at the bottom. And the one thing that I want to show is that where we saw that decrease in bacterial diversity, now we're seeing an increase in fungal diversity. But in this case, in both of these situations, we're seeing that we still see it at that 4 weeks. So here we are out at 4 weeks, and we still see in both cases that we've got a significant increase in diversity for both of these. So when you think about what this means, that we're seeing a decrease in bacteria at the beginning and an increase in fungal diversity, it could mean that we're actually seeing that we're seeing a shift in the bacterial to fungal ratio of these communities, which is a very important aspect in a soil community in ecosystem health. And so that's one of the first things that I wanted to point out. But as we start to drill down into looking at what is going on in the pathways, some of the things that we saw as far as function in these communities, we saw a difference in two main carbon pathways. One is in methanogenesis. These are microbes that are able to basically form methane through their metabolism. And the other one is pretty much the opposite, where their microorganisms are able to take inorganic carbon sources and fix those, so make them into sugars and into organic carbon. One of the things that we saw is that really this was kind of an additive effect, and so you can see that the nematodes alone in both cases, in both the Carpocapsae and the Feltier, we see that there is a decrease in the methanogenesis versus the control. So this 0 represents basically what we're seeing at untreated. So we're also seeing that when we have a pheromone, we see the same thing, but when we see both of them together, in both cases, it's an additive effect. And we see the same thing with carbon fixation, where we see that additive effect of the pheromone and the entomopathogenic nematode are actually working off of each other. So they're building a bigger response in the microbial community. This is one of the things that we saw as far as impacting just the general carbon of the system and how things are going. And One thing to point out is methane is a huge greenhouse gas. So to be able to say that you're reducing the methanogenesis in these systems by up to 35% relative to the control could go a great deal towards kind of these also building up kind of soil carbon in that system. We looked at other as pathways as well. This is showing some of the phytohormone pathways. So in this case, we're looking at both again the auxin pathways for both time 1 and time 2. So this is 2 weeks and at 4 weeks. And the thing that we saw is that for one of these, for the Carpacapsia, I'm gonna keep on murdering that name. I apologize, David. But we see that we have an increase in auxin pathways, but it's not so much where we have the nematode itself. We're really just seeing that where we have the presence of the pheromone. So we see it increased in the pheromone with the nematode as well as just the pheromone alone. And we see the decrease actually in the other nematode, in the Feltier nematode, where we see that decrease. And we actually see that it starts to recover at 4 weeks, where we've got, we still have those auxin pathways are increased at 4 weeks in this microbial community. Another one that we saw that kind of flips it around is where we saw that abscisic acid pathways in the Feltier. So Carpacapsia was increased in the auxin pathways that we see here. And here in the abscisic acid pathways, we see the same thing where the nematode itself isn't doing anything at 2 weeks, but the nematode with the pheromone in both cases is significantly increasing the pathways that we see that are responsible for abscisic acid. And we see that again, similar to what we saw before, we're seeing a little bit of an effect of the nematode itself. In this case, it's not significant, but we are seeing significant in the combination of the nematode with the pheromone as well as the pheromone itself. So it really looks like the pheromone is doing a lot to drive a lot of these plant hormone pathways in the microbial community. It's doing something to stimulate this part of the microbial community. As we look at biocontrol pathways, now these are pathways that are in the microorganisms themselves. So microbes that have that are able to have fungicide properties or nematicide properties. This is looking at the abundance of those microbes over the time in these different places and these different treatments. And here we see again that the pheromone itself is playing a big role in increasing the fungicide agents or these fungicide pathways in the Feltier, and then we see that after as we get to that second time point, we still see those effects, and we actually start to see that there are a little bit more effects. So we're seeing more insecticide agents, more basically that you're building up the natural biocontrol of the system, the microbial biocontrol. With SC, the Carpacapsia nematodes, we still see a slight increase in fungicide agents, and we really see it a little bit more at the time 2, but it's not quite the same as what we saw with the Feltier. So we are seeing pretty big differences in these 2 different types of pheromones, as well as these 2 different types of entomopathogenic nematodes. This is kind of what it looks like if we kind of spread it out a little bit to look at what's going on. We see that really the EPN with the Feltier is kind of driving these antifungal pathways. Again, it's mostly driven by where we have the pheromone. So the pheromone is really doing something to benefit this. And we're kind of seeing mixed results with the Carpicaceae. So we are seeing differences in what's going on in with these two different pheromones and entomopathogenic nematodes. As we go to actually the pathogen control, so this is looking at, we're actually counting these pathogens themselves in the soil. One of the things that we see is that in this case, before, where we saw that the pheromone was really driving kind of the microbial response and kind of the microbial pathways for some of these anti-pathogenic factors. Here we're actually seeing that it's the nematodes that are driving that. So in this case, we have these two have the entomopathogenic nematode. So the nematode itself, as well as the nematode with the pheromone. Where we have the pheromone, we kind of have a mixed result. Again, and this is for green fruit rot, the causative agent for green fruit rot. And again, with the Feltier, we see the same pattern where the nematode itself and the pheromone with the nematode, we see a strong loss in those pathogens where we don't see it quite as strong in the pheromone alone. And then with in this case with botrytis, we're seeing just this Feltier is really having the impact, and the Feltier with the hormone having the impact on botrytis. So this is just showing how the pheromone and the nematode are actually having different impacts on the soil microbiome as we kind of move through these 4 weeks of this study. So the conclusions here that we are seeing a little bit of a difference in the diversity of the microbes. We're seeing kind of an immediate loss of bacterial diversity that loss is gone by 4 weeks, but we're seeing a longer gain of fungal diversity that is still there over 4 weeks. We're seeing that there's this dynamic between how the nematodes and the pheromones are interacting with the microbes to kind of regulate the carbon cycle a little bit. And then we've kind of gone over a little bit on how the nematodes and the pheromones are interacting differently in terms of looking at the phytohormones of the plant, the auxin and the abscisic acid, as well as some of these biocontrol pathways where the pheromone itself is kind of basically encouraging the microbes to fight off pathogens, and the nematodes themselves are actually directly interacting and driving down the pathogens themselves. And so these are kind of some of the conclusions of the biology that we can make through these kind of trials that we have done. I'm going to turn a little bit of time over to Fatma again for kind of some of the larger conclusions that she has before we open this up to address some of the questions that have arisen. Fatma?
Thank you, Jacob. We were actually really excited about the results, and one of the reasons we tested 2 different nematodes and at the same time the pheromone extracts is to see what are the commons, because we knew the pheromones were common to both species. And David can tell more about the nematodes, why we select, another reason we selected 2 different type of nematodes. And one other thing I would like to mention is when we did these 2 trials, the soil biodiversity actually was really affected by the environment, and particularly the precipitation and the water. And in the fall, we had a lot less water and the rain in the field, and we think some of those effects might be relevant to environmental factor. But the common ones, we had the common effect on the botrytis and green root rot. That actually we were able to get statistically significant response. In the first study, we didn't really know whether it was the nematode versus nematode plus pheromone. Now in the second study, it actually clearly told us with the statistically significant finding that was due to Escarpicapsy, which was actually really amazing for us. And with the we are also working on a manuscript, so we will have a lot more detailed results for you very soon. And so the diversity and the water interaction was really important. And another one is, even though here the fungal diversity was shown at 4 weeks, you're probably wondering, oh, does it actually affect, what about the 6 weeks? In our initial study, we did 6 weeks, and 6 weeks, everything actually went back to normal. That was one of the reasons we thought we are not going to exceed 4 weeks. We knew some of the diversity were going to be showing as diverse. David, would you like to mention about the differences, about the nematodes?
I mean, we just chose the nematodes that would be appropriate for the targets at that timing. So Staudinemycarpicapsy has been shown to be the best nematode for control of peach borer among the commercially available ones. So we chose that. And then for the pecan weevil, actually Escarpicapsy is really good too, but it doesn't do well at all in cool conditions. So we went with S. felti, which in general is a cold tolerant nematode, though as you saw, the nematodes without the pheromones still didn't quite cause reduction in the pest population, but only with the pheromone did they control the pest.
Well, great. Well, so just so everyone knows, we need to excuse David here in a minute. He's got another engagement, but I wanted to ask one of the questions that came up in the chat to you, David, before you leave. The question is, what occurs after the beneficial nematodes control all the pests you want? Do they die off or find something to feed on we do not want?
For the most part, these beneficial nematodes have little or no effect on most beneficial species. So we've shown that they don't do much to the lady beetles or ground beetles, and that, especially in comparison to what they're doing to the pests, and especially in comparison to chemical pesticides, which will take out all the natural enemies as well as the pests. So the nematodes, I mean, they will stay in the target area maybe for years, but the population levels won't normally be as high as they were when you first applied them. So you don't expect a lot of, you usually have to apply the nematodes every year for that target pest. There are some exceptions where the nematodes will persist over years. We don't, in most biocontrol cases, you don't expect the natural enemy to wipe out the pest population. There's usually going to be some left over, but then the nematodes that are remaining may find another host. So if you're targeting the pecan weevil and they take that out, there'll probably be a few left, but they may go to white grubs or something else like that in the soil.
Fatma, do you have any comment to add on that?
No, that was a question actually I have been asked a lot. And so David was one of the first person I asked, and there were a number of actually other entomopathogenic people who work with entomopathogenic nematodes, and I said, well, does it attack anything else? And there are very many studies. We actually were asked in the grant proposal, and there are a lot of studies have been done with the entomopathogenic nematodes that they don't harm the beneficial organisms. So for that aspect, we were very lucky.
So here's another question for you, David, before you leave. Do the entomopathogenic nematodes have the capacity to be undetected for the host in the soil? If that's so, do you have any hypothesis on how they're able to remain undetected by the host?
It's a really good question. There has been some work on, if you're talking about the insect pests detecting the presence of the nematodes, there has been some evidence that some insects can detect them, and they have various defenses, like they'll try and groom the wet nematodes off of them. Well, of course, once inside, they have their internal host defenses. But for the most part, the nematodes, they're queuing into that pest when they're in the soil, based on various volatiles that insects are releasing, and they will get to them. So, you know, usually the insect is unaware and will be attacked, or sometimes they're in a pupal cell and they're not going anywhere, and the nematodes will get them there. But for the most part, the insects have not been shown to avoid the attack, because probably they can move faster than the nematodes. So I got to jump off, but I really appreciate it, and people can send me more questions. Thank you very much.
All right, thank you, David. We appreciate your time and your expertise on this call. So we've got another one that's specific for you, Fatma. In which formulation are the nematodes applied? How are they formulated and applied in the field?
Oh, that's a very good question. The nematodes, we don't do nematode formulation, and our plan is to use the commercially formulated nematodes. The field trials have been done using the nematodes that are produced at the USDA. And in our case, the pheromones are formulated vegetable powder, so they're a dry powder.
Perfect, thank you. And then, so there's the question, how are you quantifying the pathways? Are you using copy number variants from the 16S data, either proprietary or an open source tool? So yes, we're basically using the copy number, ITS 16S copy number, to look for changes in abundance of the microbe. So we are able to do relative abundance of those as they change. And then we've got a kind of proprietary algorithms where we're able to check it back against fully sequenced genomes, and we're able to kind of know what 16S sequences correspond with what function, or ITS sequences. Fatma, I think this one is one for you. Do you have any hypothesis on how nematodes control pathogens?
Oh, that's a very good question. Oh, I don't know how they would promote, in this case, actually their abundance. That was a very surprising result for us too. We really didn't expect, particularly the amount we put in the soil was so minute that, you know, its effective even when we are treating the nematodes is very low. And by the time it gets into a field, because we have a huge dilution there, and we really didn't think there would be anything, that was a very surprising effect for us. We don't know how what mechanism it's using to promote the abundance of those really great biologicals.
Yeah, and I think part of it is that when you are looking from a biological aspect, you are kind of trying to address the system. So if for example, the nematodes are helping to make the plant healthier, and the plant then helps the microbiome to be healthier, and the microbiome, part of the healthy microbiome is to fight off pathogens better. So it might just be that you're kind of fixing the whole system rather than there's a direct impact from the nematodes themselves.
That is actually what we thought. It may not be a single thing because we directly target the ecosystem. And if they have supportive relationships among different fungi or bacteria, we didn't really know. So, but these can be studied actually individually, one at a time. But again, because we are targeting the whole ecosystem, we did think about the plant aspect because that might be, you know, summer versus winter, and these are actually around the trees. Even though we didn't mention about the trees and how they, you know, act spring versus fall.
So I think we've got time for one last question, Fatma. I'm going to ask you this because I also want to know the answer for this one. Are you contemplating long-term studies to evaluate the effects on the biodiversity of the microbiome of the specific soil due to the effects of the nematodes and pheromone applications?
Yes, we are interested because we know the short term, but we are also interested in long-term effect, how that would improve the soil effect. It's definitely one of our priorities.
All right, well, and to those of you that we did not get to your questions, we will make sure to respond to those and send you those via email. Fatma, again, thank you so much for being on this webinar with us, and we'll thank David again. And thank you very much. Thanks for all those who attended, for the questions, and we look forward to hearing from you soon.
Thank you for inviting. We really enjoyed it.